Hello everyone, this is Katish here. In this video, I'm going to go through the AQA 2022 Combined Science Biology Higher Tier paper question number 3, which is on cell biology. Amylase is an enzyme that digests starch. Which organs in the human digestive system produces amylase? So you have liver, small intestine, and large intestine. Liver doesn't produce amylase. Remember, amylase is responsible for breaking down carbohydrates. Salivary glands, yes. Stomach, no. Liver, no. Salivary glands, pancreas, and small intestine, yes. That is correct. So let's mark that. That will be number 3. And stomach, pancreas, and large intestine. Large intestine doesn't produce enzymes in terms of digestion. A student investigated the effect of pH on the activity of amylase. This actually is referring to a required practical on enzymes. Required practical on enzymes. This is the method used. Prepare amylase solution at 5 pH or pH 5. Mix the amylase solution with starch in a boiling tube. Remove a drop of the amylase starch solution every 30 seconds and test it for the presence of starch. So if you have a test tube with pH 5 amylase, right now amylase is actually the enzyme. The pH 5 is actually a buffer solution. You don't want to change the pH, and what you're trying to do is add amylase starch mixture as well. So you have starch as well. Now what happens is the starch is going to be broken down by amylase. So starch is broken down by amylase to form glucose. Record the time when all the starch has been digested. Now starch will go blue-black with iodine. Iodine, it will be black. With glucose, it will just simply be the orange color or brown color. Orange color. So what is the independent variable in this investigation? The independent variable is what you are trying to change. The student investigated the effect of pH. So the effect of pH would be the independent variable. So the pH of the amylase solution or the buffer solution. Now as a quick note, right, the main organs that produces the enzymes in our body or digestive enzymes. In the mouth, you get the salivary amylase. Stomach is pepsin is produced. The small intestine produces the amylase, trypsin, and lipases. And the small intestine also produces the maltase, sucrase, lactase, and peptidase. Question 3.3. Describe how the student would know when all the starch has been digested. Now, remember, iodine is used to test for starch. So, iodine solution will not turn blue-black, or the iodine solution will just simply stay in the orange-brown color. 3.4. Figure 6 shows the student's results. So, remember, it's the same pH. Or it's actually the pH is changing. So, for pH 5, 6, 7, and 8. And the different times it takes for the starch to be digested. What is the optimum pH? The optimum pH is going to be at this point over here, and that is 6.8. The least time it took for the starch to be digested. A scientist did a different investigation. This is the method used. Prepare amylase solution at the optimum pH. So that would be 6.8 pH. Mix the amylase solution with starch in a boiling tube. Measure the concentration of sugar every 10 seconds for 2 minutes. So what they're actually trying to find out is the rate of digestion of this starch. So concentration of sugar should actually go up as it's digesting more, but eventually when all the starch has been digested, then the concentration remains the same. Remember, the enzyme is not going to be used up by the end of the reaction. How much time did it take for the amylase to digest all the starch? Now that's happening when it's a flat line. Now the flat line looks like 80, but I think it's definitely at that point, definitely at this point there. So I would say 82 seconds. So question 3.6. Determine the rate of sugar production per minute at 40 seconds. So looking at the graph, you would need to draw a tangent at 40 seconds. Now, because they're asking for a minute, I'm going to draw a line right up to 60 seconds. So let's see. That should be about 60. The line should actually be slightly be above. There you go. Right. So the difference, remember the gradient is going to be the change in the y value divided by the change in the x value. That's 4.3. 4.3 minus this would be 1.9. 1.9 and 60 seconds. So that would be, because it's per minute, I don't really have to divide. So 4.3 minus 1.9, that should give me 2.4. 2.4. I'm not dividing it by 60 because it's per minute. And if I divide that by 60, I'll have to multiply it again by 60 to get it in per minute. So the answer is 2.4. So rate equals gradient, which is delta y over delta x, which is 4.3 minus 1.9 divided by 60. Multiply that by 60 again to give me, that should give me 2.4. 2.4. Any answer within the range of 2.1 to 2.5 is actually acceptable here. Question 3.7. Explain how the structure of enzyme molecules is related to the effect of pH on the activity of amylase. So this is referring to the lock and key mechanism. Just to review a little bit on this, let me give you a bit of notes and then discuss the answer. So remember, enzymes have a very specific shape, which is a 3D structure. So if that's the enzyme, they have a very specific shape called the active site. So this could be the amylase. and the substrate will have the complementary shape, right? Which is the starch as a complementary or the opposite shape. Now, if there's the pH increases, too much pH, or the pH change, then the active site is destroyed. Right? And we call that the enzyme is denatured. Now, when that happens, the substrate cannot actually fit into the active site, and the enzyme will stop working. Now, usually at low pH, this change is reversible, but at very high extreme pH changes, then that change is irreversible. So, the answer for the 6 marks: enzymes are protein molecules that have a 3D structure. They have an active site which has a specific shape. The shape of the active site will only match the shape of the substrate, like a lock and key. Starch is the substrate for amylase. At pH values above or below the optimum, the shape of the active site is changed. So the substrate can no longer fit the active site. At extreme pH values, the enzyme is denatured. So the shape of active site is changed and amylase can no longer digest starch. This causes the rate of digestion to decrease. That's the end of question number 3. I will see you in question number